Hey guys, so welcome back. We're gonna kind of finish up this next section. This next section is gonna go into bacteria and talk about how they get energy and release energy, and then we're gonna get into how they reproduce a little bit, kind of talk about that. So, um, one of the things is we talked about before that we can look at their cell shape and identify them, and a lot of times from their cell shape we can know the species, but most of the time we got to stain them, gram stain them. And sometimes gram staining them, we can tell exactly what species we're talking about. But then other times we really don't know. So then we got to talk about, okay, how do they use energy? How do they release energy? What do they use for energy? And there's all kind of little tests that you can do. So if you take microbiology, you definitely can. You definitely learn all those. So again, they fit two categories. Either they're autotrophs, they make their own food, or they're heterotrophs. They got to get their own food. Okay. So if they're autotrophs, they're either photoautotrophs or they're chemoautotrophs, meaning that they're going to do some type of photosynthesis without a chloroplast, um, or they're going to do chemosynthesis, which they're going to take chemicals in. And again, we talked about the hydrothermic vents. That's them down there. If they're heterotrophs, then they're going to use chemicals, chemoheterotrophs, or they're going to be saprophytes. Most of the bacteria fall in a saprophyte um, category because they feed on dead and dying things, and they're decomposers of the world. Okay, so uh, going on. So the autotroph ones, the ones that we want to talk about are the cyanobacteria, and a lot of times these are called the blue-green algae before. Again, they were in the algae group, and sometimes the algae group, going back to chemus, might be in their own kingdom because the algae group kind of defy being a plant. They kind of defy being a protist. So again, it's up to you where you want to put them. I'm going to leave for my sake. They got enough plant qualities. They're going to be plants, and if they're more single cell qualities, then they're going to be protists. I'm not going to get real hung up on that. Okay. So typical cyanobacteria, you kind of look at it. They've got thylakoids in here, but they're not like in a chloroplast. They're not structured. Okay, so they're very different than um, what a plant has, but very similar in the process that they use. Okay, how it works and how it goes through. Okay, and you can kind of see how they're very, very different. The blue green algae usually are good tellers that the water you probably shouldn't drink. Okay, it's just they're not, and they're very obvious when they say they're blue green. It's a very obvious color. If you've ever seen them or ever get to see them, they're very obvious out in the environment. Okay. So we talked about the cyanobacteria. So the chemoautroph, the chemosynthesis guys, are the guys we talked about down in the hydrothermic vents. And again, you can kind of see what they're doing at the bottom. They're using hydrogen sulfide, and that's coming up from these vents, very hot vents. Again, remember from physical science, I think we talked about tectonic plates a little bit. If you've done earth science, the majority of the volcanoes are under water. They're in the Pacific Ocean. They're underwater. So this is what's going on down there. You can see the different thermal vents. They're producing sulfur, hydrogen sulfide, and the different compounds. And they're picking it up and they're breaking it down. And so they provide the basis for this food chain. It's not a plant. Again, this defies that when we were talking about ecology that all food chains start with plants. No, they don't. In this food chain, it starts with a bacteria that's chemosynthetic. So just when you think that you know it all, here we go down here, and oh my gosh, look, this is going on. So it's, again, hello. Just when we think we know it, we don't. And the only one really, again, who can enjoy this is God. He gets to see it every day. We have to, again, take a remote control apparatus and go down there and look, or a submersible. If you guys have ever been to the Titanic exhibit over in Gallenberg, uh, well, Pigeon Forge, and they talk about the submersible going down. We actually got to meet one of the captains who went down and got to see the Titanic, and it was unreal how long it took. Again, because you got to take your time because you don't want to get the bends going down or get the bends coming up as you go through. And I think In physical science, we talked about that. We talked about how pressure differences and what's going on and in that. So that all relates back to challenge one to physical science. But so if you're going down and you can't stay that long, so it's like you travel down, I don't know, something like you said, 3 or 4 hours. You're only down there an hour, and you got 3 or 4 hours back. So that's like almost a whole day, and you have to hold it. You can't go to the bathroom. Okay, there's no toilets on the submersible because it's all little. Um, so very interesting talk by the fellow. So again, if you go to see the Titanic exhibit, they'll talk about that. So they won't talk about the hydrothermic vents, but they'll talk about where the Titanic was found, and you get to see what what it would take to get down there in a submersible. And this is even further down. So again, all the Meg movie, no, not true, not true. Okay, none of that's true. This part is true. This part right here, we're talking about the hydrothermic vents. Oh yeah, this is true. These two berms right here that you see on the coast, when you're walking out on the seashore, you may get to see some tube worms. They're like maybe this big. Down there, they're like 6 foot. Okay, because they don't have any um, have any worries. They live in the bottom of the ocean. They have plenty of food. So again, very interesting place to live. So that's those guys. Um, so getting and releasing energy. So we talk about the heterotrophs. So let's talk about a few of these guys. So here is the nitrogen cycle, and we talked about the nitrogen cycle back in module 3 with ecology, and so this is what's going on. So the prokaryotic guys are taking atmospheric nitrogen, and they're putting it in a usable form, nitrates or nitrides, for plants to absorb. And then again, so then the herbivores eat the plants, and then we eat the herbivores, and that's where we get our nitrogen. And if you remember back, what's the only macromolecule? Well, there's two macromolecules that contain nitrogen. So what's the two macromolecules that contain nitrogen? Okay, so it's not carbohydrates, it's not lipids, nucleic acids, DNA and RNA, but then proteins have nitrogen. And so and proteins are important. So this is that how we get our proteins. So that bacteria absorb it from the atmosphere because we can't do it, plants can't do it, but they can do it. So very important to the nitrogen cycle are plants. So again, majority of bacteria are good guys. Okay. Um, so let's talk about the types of respiration they can do, because there's three basic types here. They can be obligate aerobes, and we talk about obligate. If you're obligated, what does that mean? Okay, what does it mean to be obligated? Hopefully, you're saying to yourself, if I'm obligated, I got to do it. There's no ifs, ands, buts. I got to do it. Okay. So if you're an obligate aerobe, you got to have oxygen. If you're an obligate anaerobe, you can't be with oxygen. You can't be in the presence of oxygen. We'll talk about endospores here in a minute. But then you got some guys who can live in both, and they're facultative. Okay, so when we're looking at our obligate anaerobes, these guys, because they can't live in the presence of oxygen, what they'll do is when exposed to oxygen, they'll wrap themselves up in this thick inner wall, and they'll produce a thing called an endospore. And so then they can survive um, during this process. So if they don't have oxygen, they become endospore, and then when they got oxygen, then they'll pop out and be and live again. So obligate anaerobes have that ability. This is indicative of the guys that we talked about, just mentioned a little bit ago, the Clostridium botulism, Clostridium tetani, um, the one that causes gas gangrene. And what happens is, like you pour in hydrogen peroxide into a wound and it starts to bubble. Well, that'll kill them. Um, and that's one of the things that why hydrogen peroxide is so good when you as an antiseptic. Okay, and this is why botulism can live for quite a while in improperly canned foods. Why your foods have to be canned properly, or when you cook them, you got to cook them till they're boiling for like 5 minutes. and that'll kill the endospores. They'll kill the bacteria. Okay, so when we look at them and compare both sides, you can see here that the obligate aerobes definitely need oxygen, and the anaerobes need to live in without oxygen. You can see the different types of uh, bacteria both sides. And again, it doesn't really matter, these guys that are aerobes are good guys, these guys that are anaerobes are bad guys. No, they're all dispersed. Okay, so it just depends on what bacteria we're talking about. And again, we'll be able to isolate them and know which ones we're talking about. Okay, so that's one view there, and then here are them growing in a petri, not a petri dish, but this is a auger test tube. And the obligate aerobes like to be at the top because they like oxygen, but the obligate anaerobes are down here at the bottom. And so if they're facultative or in betweens, they'll grow all the way through it. And so that's another way to tell what kind of bacteria we're dealing with. Okay. Um, then we want to talk about the conditions for bacterial growth, and there are five conditions. And again, if you think about it in your mind, think about like what causes bacterial growth. What do you know already in your brain? What do you know? Well, number one, we know that everything needs nutrition, so they got to have food to eat. Okay, so when we're looking at growing bacteria, you grow them on an agar plate, which is nutrient agar. Some of them are real specific, so some of them won't grow on that. Some of them you'll have to add something to it to get them to grow. But they need food. They need a moderate temperature, and so 27 degrees Celsius to 30 degrees Celsius. So anywhere from a little bit above room temperature, so about 70, a little bit above 70 degrees, so 80 to 100 degrees. That's why bacteria love warm places. So like in the summertime, if you leave stuff out, it quickly bacteria from the air falls into it, and quickly you can culture some bacteria. They like moisture. They need moisture to grow. Um, Not too wet, but not too dry. They don't really like dryness. They like darkness, but some may need limited light. Um, and then again, we talked about the proper amount of oxygen, whether they need it, don't need it, or can live with both of them. Okay, so those are some things to think about when we're growing bacteria. Um, so bacteria are interested in reproduction, and when we're talking about basically, we've mentioned this again in module 1, and we talked a little bit about again, I think, and again in module 3, no 4. So we're talking about reproduction. The thing about bacteria is most of the time they're asexual reproduction, so which means they come from one parent, if you will. One bacteria splits into two, and they're identical, and so that pretty much is a binary fission. Okay, and within 20 minutes, one bacteria becomes two bacteria, and two bacteria. So in 10 hours, one bacterium could be multiplied into a billion. Okay, but again, when we're talking about that, we've got to go back and talk about our ecology chapter and talk about carrying capacity. There's only so much bacteria can hold in. Uh, so if I got a pizza or whatever, there's only so much that it can hold, and then it's going to be done. So again, you're going to get this growth spurt, and then it's going to come off because it can't live there anymore because of everybody's eating and reproducing and having waste, and they just can't survive that way. Bacteria do sexual reproduction, but when we talk about this is not trade in sperm and egg. This is not an ovary and a testes. This is not an ovum and a sperm. This is very different. But in the terms of sexual reproduction, we get a new genetic information. Then yeah, okay. So it's a type of reproduction. Conjugation, where two organisms join for the purpose of DNA transfer. Okay, and this is you can see the infographics there for those in your textbook. So when we look at them, let's see, I think I got an infographic here too. So when we look at this, as we look at what's happening, it's conjugation versus this is conjugation where they just exchange information. So in the donor, they have an extra plasmid, and so they'll replicate their plasmid and they'll share it with the other donor. So both of them have now the same information. This one, the recipient's different. Now this, again, for the sake of sexual reproduction, with the basic idea is that you get new genetic information. Yes, there's new genetic information there. Okay, binary fission just means that they split in half, they replicate the plasmid, and they split in half, and it's the same. Okay, so when we talk about conjugation and binary fission, we can talk about a couple other ways that they can reproduce, and this is transformation and transduction. So the thing about bacteria is, and we've talked about this, is that bacteria, they're very easy to pick up information. So if a bacteria cell dies, let's go back and look at the definitions because these are two vocabularies. When bacteria dies, then it can pick up that information from a live one can pick up information from a dead bacteria. So that's the first one. So transformation, the transformation of DNA segment from a non-functional donor, a dead bacteria, to one that's a functional one. Okay, so that's transformation. We saw that back in DNA. We talked about Fred Griffin, and we talked about how he figured out that it was DNA that was being transferred over. And then we talked later about, let's say, Avery, McLeod, and MacArthur did the transduction one, where the virus gave its DNA to a bacteria. Okay, so a virus gives DNA from one to another. Okay, so that's the difference between the two. So we'll look back. So here's transduction, our transformation. So we get this bacteria sitting here, and then we're going to insert this plasmid, and we're going to get this transformed bacteria over here. And now it's going to be antibiotic resistant. That's what we see happening a lot of times. Like, and again, like if you don't take your antibiotics all the way to completion, like you feel better within 5 days, and so you've got 10 days of antibiotics, you just don't take the next 10 days. Well, all the bacteria that are dead are there that died because they're antibiotic sensitive. And you got all this bacteria right here that's alive, will take that information to themselves and be transformed and maybe possibly be antibiotic resistant to the next antibiotic you may try. So again, that's where we're seeing antibiotics. I'm not saying that's going to happen to you every time you do that, but I'm saying that's why the doctors always tell you to take all your antibiotic to make sure you kill all of the bacteria. But bad news is you kill all of the bacteria, even the good guys, when you take an antibiotic. So because antibiotics aren't specific for that. So you see transduction is the whole bacteriophage, the virus injecting in DNA into the bacteria, and then transformation, they pick it up, and conjugation, another bacteria actually gives it to the other bacteria. Okay, so that's the basic. Again, the underlying principle is I hate using the word term sexual reproduction, but the underlying principle is that all three of these have new information, new genetic information, and that's the basics of that. So when we look at that, okay. So again, this is another one of those things I like about our book. It says the idea of bacteria are very important. So when we think about it, how are they important? And again, we talked about this. They make vitamin K, they make vitamin B or vitamin Bs in our intestinal system. They keep us, even our bacteria on our skin is normal flora, and so it keeps everything normal. That you need a certain amount of them. But again, when you take an antibiotic, it kills everything, kills them all. That's why also too they'll ask you to take, eat yogurt, take a probiotic, so that puts back your what they call your normal flora, your normal bacteria. And then they talk about what about in a biosphere, and that's the part. If you read about it, that's we try to create this biosphere too. We put all these people in the biosphere, and it was kind of the precursor to sending them to Mars and terraform Mars or whatever. sci-fi thing you want to think about. And so they're in there living, and they miss putting the bacteria in. And because the bacteria is not in the soil and the bacteria is not in our bodies and the bacteria aren't there that are needed, it was not a success. And so even the little guys are important in God's kingdom. And that's important to remember, that he set this up and everything is going on as he's designed it to work. And when we don't have them, then things don't work like they should. Okay. So let's look at some questions on our own. And we talked about saprophytic bacteria. Can saprophytic bacteria be autotrophic? And that's unless they're facultative, right? Bacteria, then they can be and not be. So anyway, can aerobic bacteria be chemosynthetic? And we go back and kind of look at that. And then we talked about how does bacteria reproduce and exchange genetic information. And again, we talked about the basic form of asexual reproduction. It just divides and splits in two, and by 8 hours, 10 hours, we have a billion of them. Or they could pick it up from a virus, or they could pick it up from their environment, or they can actually exchange information or get information from another bacteria. So that's pretty much it for this section. We're going to look at the next section and talk about it and get a little bit more specific in nature. So, but this is for all for now, because we went through the on your own questions. So double check and go back through and get the basics again. We're not looking for you to be have PhD in bacteria. Okay, we're just giving you some basic information, and then we're gonna kind of tie it to this next part that we're going to talk about. Okay, thank you again for watching, and see you in the next part.